Hello everyone, thank you for paying attention to our work published in Cell Regeneration about how Rif1 regulates mouse embryonic stem cells' fate potential through interacting with non-canonical polycomb repressive complex PRC1.6. I'm Jiale Chen, one of the co-first authors of the paper and currently a graduate student in Kaiyuan's lab at Xiangya Hospital, Central South University. Stem cells are a class of cells that can self-renew and can be induced to differentiate into various embryonic tissues and organs such as hematopoietic cells, blood vessels, nerves, and the heart. The study of embryonic stem cells is of great significance for both basic research as well as regenerative medicine. During early embryonic development in mammals, the differentiation potential of cells is becoming increasingly limited as a development process. In mice, cells in fertilized eggs and two-cell stage embryos are totipotent, which can develop into complete individual organisms, including the three germ layers and the extra-embryonic tissues.
While embryonic stem cells only have pluripotency.
Pluripotent stem cells can only differentiate into cells of three germ layers. Study of totipotency can help understand human embryonic development.
However, limited by material scarcity, mechanistic studies of totipotency are largely impeded. Some researchers found that MERV, a ritual transposome, was highly expressed at the totipotent stage, which is a two-cell stage, during mouse embryonic development.
In 2012, it's reported that a small part of embryonic stem cells induced pluripotent stem cells cultured in vitro did not express the pluripotency marker OCT4 but highly expressed MERV. The stemness of this small part of cells are explained. It has totipotent properties and can develop into embryos and extra-embryonic tissues. Because the transcripts of these cells are very similar to transcripts of the two-cell stage of embryonic development, this small group of cells highly expressing MERV is called two-cell-like cells. This provides a sufficient cellular basis for the study of totipotency. There are also various reporters applied to genome-wide siRNA screening or CRISPR-Cas9 screenings. They are respectively designed by Yuhan Lu Group, Yizhang Group, Maria Elena Group. Genome-wide screening aims to generate and screen a population of mutant cells so that it could identify genes involved in regulating the fate potential. Most of the factors screened which were overlapped with our results, like Rif1 and PRC1 complex. In their screening results, many subunits in the PRC1.6 complex, such as Pcgf6, Rif2, RYBP, MGA-Max, can significantly increase the proportion of two-cell-like cells in mESC, indicating that the PRC1.6 complex is also indispensable for the control of totipotent state. Since MERV is highly expressed in two-cell-like cells, a cell line using MERV-LTR to drive the red fluorescent proteins tomato could be used to screen for genes regulating the transition of embryonic stem cells to two-cell-like cells. In an SHR screen target genes regulating the reprogramming of mESC into 2CLC, we found that the phenotype was most pronounced when Rif1 was knocked down. In addition to Rif1, we also found that knockdown of subunits of the PRC1.6 complex, Rif2 and Pcgf6, MGA and MAX all increase the proportion of red fluorescent cells. Rif1 is a multifunctional and evolutionarily conserved gene which were first described as a Rif1 interactor involved in transcriptional silencing and telomere regulation. Latest studies found that it is also involved in telomere length maintenance DNA damage response during replication. However, it's still not clear how Rif1 functions in concern with other totipotency factors to regulate the cellular plasticity of embryonic stem cells. To determine the position of Rif1 in the regulatory network that controls the fate potential of mESC, we first summarized previously reported factors that regulate regulate the reprogramming of mESC into 2 CLC, including 21 repressors and 4 activators. Next, we mapped the protein's interaction network of these 21 known repressors. It can be seen that proteins belong to the same complex or participate in similar post-translational modification pathways such as simulation and methylation are clustered together to form distinct functional modules. However, the results also show that among the 21 verified repressors, Rif1 only has a potential association with the histomolecular chaperone CARF1A. We collect and analyze the RNA-seq data after the deletion of these 21 repressors. Firstly, we performed the PCA analysis and found that differentiationally expressed the genes after silencing Rif1 and the subunits of the PRC1.6 complex, PCGF6 and RF2, which could be clustered together because the repetitive elements of the genome also have a great impact on the stimulus potential of embryonic stem cells. In addition, we performed the same analysis on repeat elements and found that Rif1 silencing could be clustered together with the subunits of the PRC1.6 complex, PCGF6, RF2, and the differentially expressed repeat elements after MGA silence. Our correlation analysis heat map also showed that Rif1 silencing with PRC1.6 subunits, PCGF6 and RF2, and MAX and MGA silencing differentially expressed genes and repeat elements showed a clear correlation, indicating that Rif1 is closely related to PCGF6. Similar genes and repeat elements are regulated by RF2, suggesting a functional link between Rif1 and the PRC1.6 complex. We also draw a Venn diagram of up-regulate genes and the repetitive elements after deletion of the three and found that they all overlapped to the same extent. Analysis of repetitive elements showed that the overlap was even more significant. More than 19% of the repetitive elements regulated by PCGF6 or RF2 were also evaluated after Rif1 deletion, that is, repetitive elements whose expression was upregulated after RF2 and the PCGF6 deletions were also regulated by Rif1. The analysis of transcriptome dynamic pointed to a functional link between Rif1 and PRC1.6. The PRC1 complex is heterogeneous, containing several subtypes according to distinct molecular compositions. To interrogate whether the connection of Rif1 to the PRC1 is specific to the PRC1.6 complex, we collect and analyze the transcript data from the mESC with single knockout of Rif1, PCGF1 or PCGF6, double KO of PRC2 and 4 or PCGF3 and 5, and triple KO of PCGF1, 2, 4. Both the transcriptional changes of the genes and the repetitive elements demonstrate a specific correlation between Rif1 and PCGF6. We further performed geo-enrichment analysis on these differentiate genes induced by the deletion of Rif1 or the PCGF proteins. The results revealed that deletion of Rif1 or PCGF6 influenced similar site of genes enriched in cell junction assemble development as well as multi-meiosis processes, which were markedly different from influenced by the other PCGF proteins. To further verify this conclusion, we used the LacO-LacI system and to study the interaction between them. The results showed that the Rif1 did not overlap with the red fluorescent in cells co-transfected with DS-red LacI PCGF125 fusion proteins plasmids. That is, GFP Rif1 had no LacO-site aggregation in the cells co-transfected with DS-red LacI PCGF6 diploid fusion proteins particles. The green fluorescence of Rif1 has a permanent bright spot, which is highly coincident with the red fluorescence. It further proves that Rif1 interacts with PCGF6 and not with PCGF125. To explore whether there is a physical interaction between Rif1 and PCGF1.6 complex, we first co-transfected 293 T cells with plasmids expressing HA-tagged Rif1 and flag-tagged PCGF proteins and immused using anti-flag affinity gels total participation. It was found that HA Rif1 was only detected in flag PCGF6, indicating that among the 6 PCGF proteins, Rif1 only physically interacts with PCGF6. Through the previous experiment, we confirmed that the interaction between Rif1 and the PCGF1.6 complex is mainly mediated by PCGF6. Rif1 contains 2 functional domains, a hit-like repeat and a C-terminal domain. Next, to explore the functional domain of the direct interaction of Rif1 with PCGF6, we used the different Rif1 truncation mutants that had been contrasted to determine the domain of the direct interaction of Rif1 with the PCGF6 subunit. Again, by the LacO-LacI system, we found that both N-terminal and the C-terminal regions of Rif1 are involved in the interaction with PCGF6. We also confirm that Rif1 also interacts with other subunits of PRC1.6 complex, but from the fluorescent statistics, we can be seeing that interaction between Rif1 and PCGF6 is the strongest, indicating that the interaction of Rif1 with the PRC1.6 complex is mainly mediated by PCGF6. We next examined the interaction between endogenous Rif1 and the PRC1.6 components using cell lines that were knocked in HA tagged in endogenous Rif1, MGA, and PCGF6 in mESC. Our IP results show that endogenous Rif1 interacts with subunits PCGF6 and RF2 of the PRC1.6 complex as well as MGA. To further verify this conclusion, we used shRNA to knock down other subunits of the PRC1.6 complex, and the results show that PCGF6 still showed a strong interaction with Rif1 after other subunits such as RF2 were knocked down. Other subunits of the PRC1.6 complex do not affect the interaction of PCGF6 with Rif1. The PRC1.6 complex composed of many subunits, and the integrity of the PRC1.6 complex is essential for its transcriptional regulatory function. To investigate whether Rif1 is a novel component of the PRC1.6 complex, we first examined the assemble of the PRC1.6 complex after knockdown of Rif1. Co-immunoprecipitation analysis of RF2 and RYBP with endogenous HA-tagged PCGF6 showed that RF2 bound to PCGF6 was significantly reduced after Rif1 knockdown, suggesting that reduced Rif1 expression impairs the interaction among these subunits, which indicated that Rif1 may help maintain the integrity of the PRC1.6 complex in mESC. We then used molecular sieve chromatography to verify the integrity of the PRC1.6 complex after Rif1 deletion and explore whether Rif1 is a new component of PRC1.6 complex. The results show that in the control group, Rif1 co-eluted with RF2 and PCGF6, while in the Rif1 deleted group, Rif1 was not detected, and both RF2 and PCGF6 were eluted later in the nuclear extras of the control group. Smaller fractions were detected, indicating that the deletion of Rif1 may cause the partial disruption of the integrity of the PRC1.6 complex. In addition, part of this reduction may be caused by the deletion of Rif1 itself, indicating that Rif1 may be a new component of the PRC1.6 complex. To explore the potential function of the PRC1.6 complex, we first analyzed the distribution of Rif1. Motif analysis revealed that the Rif1 bound peaks was also enriched in the constant binding sequences of the PRC1.6 components, MAX and E2F6, suggesting the co-occupies of Rif1 and the PRC1.6 complex. Approximately one-third of the Rif1 peaks were also occupied by PCGF6, and the overlaps between Rif1 and RF2 or H2AK119 ubiquity slightly less than that of PCGF6. To investigate if Rif1 deletion compromises the genomic targeting of the PRC1.6 complex, we analyzed the distributions PCGF6, RF2, and H2AK119 ubiquity on the Rif1 bound region control and Rif1 downregulated mESC. We observed that the binding of PCGF6 as well as RF2 to the Rif1 bound regions were markedly reduced after the downregulation of Rif1, suggesting that a subset of genomic distribution of the PCGF6 complex was dependent on Rif1. Since PCGF6 is the characteristic subunit of PCGF1.6 and the interaction of the PRC1.6 complex with Rif1 is mainly mediated by PCGF6, we focused our analysis on genomic regions co-regulated by Rif1 and PCGF6. We annotated the Rif1 and PCGF6 co-occupied genomic regions that showed decreased PCGF6 binding upon Rif1 knockdown, identified 3,626 corresponding genes. A 2C gene list consists of 869 genes were generated previously. According to their upregulation at the 2C and the zygotic genome activation stage in mouse embryonic development, we found that the 2C genes were significantly enriched in the Rif1 bound regions that showed decreased PCGF6 binding upon Rif1 knockdown. To investigate the cooperation of Rif1 and PCGF6 in regulating the fetal potential of the mESC, we performed an RNA-seq analysis with mESC downregulated of Rif1 or PCGF6. The gene cycle enrichment analysis revealed that the 2C genes were significantly upregulated in mESC with reduced expression of Rif1 or PCGF6. We further examined the expression of all the 2C genes using RNA-seq data and identified our different clusters. We uncovered 195 genes in cluster 2 whose expression were consistently upregulated in mESC, downregulated of Rif1 or Pcgf6. Interestingly, genes in cluster 2 displayed higher Rif1 and Pcgf6 binding, and the amount of the Pcgf6 on these genes were markedly decreased in mESC deleted of Rif1. We noticed that several 2C stage marker genes such as Zfp352 or Zscan4 were in cluster 2. The transcription activation of repetitive element MERVL is a marker for the 2C-like totipotent state, and the 2C TD tomato reporter can reflect the transcriptional activity of the long terminal repeat controlling the MERVL expression. Consistent with our previous study, the knockdown of Rif1 expanded the number of TD tomato positive cells. Knockdown Pcgf6 but not the other Pcgf members also significantly increased the TD tomato positive cells. To further examine the cooperation between Rif1 and Pcgf6 in regulating MERVL, we knocked down Rif1 and Pcgf6 using shRNA and compared the effect from individual knockdowns. While knockdown of Rif1 induced more TD tomato positive cells than that of Pcgf6, the double knockdown showed no addictive effect, suggesting that Pcgf6 were in the same regulatory pathway as Rif1. Additionally, we analyzed the expression of MERVL using our ChIP-seq and RNA-seq data and unambiguously identified 66 Rif1-bound MERVL loci that were transcriptionally activated after Rif1 deletion. 48 of them could also be activated by Pcgf6 knockdown, indicating that these MERVL loci were co-regulated by Rif1 and Pcgf6. Taken together, these results suggest that Rif1 and the PRC1.6 complex could form a functional module to control the transition from pluripotency to totipotency by restraining a group of 2C genes and MERVL elements. Our study reports a novel interaction between Rif1 and the non-canonical PRC1.6 complex. Moreover, Rif1 contributes to the genomic target of PRC1.6 to a group of loci and the deletion of either Rif1 or Pcgf6 activates many 2C genes and MERVL, suggesting that Rif1 PRC1.6 module plays an essential role in controlling the cell rate potential of mESC. I want to thank my supervisor for his guidance in this work, and thanks for your attention to our work. I hope you could continue to keep a watchful eye on the content of cell regeneration. Thank you.